All right, so we're going to start lecture 2, and this is in relationship to animal diversity. Well, I guess I shouldn't say lecture 2. It'd actually be like lecture 11. But this is in regards to this next series of lectures dealing with animal diversity. And so we've already covered what an animal is. We've already covered some of the evolutionary ideas that led to the animals arising. And we talked about the most simple animals: sponges, jellyfish, coral reefs, and sea anemones and hydras. And so we talked about this group over here. We had no symmetry, we had no tissues for sponges. They were filter feeders, and they were also known as parazoa, which is beside animals. And then remember, everything underneath this bar is eumetazoa, or true animals. So our first true animal, even though they're all animals, true animals in the layperson's sense, the jellyfish were radiated in their symmetry. So they gained some symmetry that wasn't seen in the sponges. And now we're going to be moving into this next group of animals. We also started seeing tissues. This is where, so the idea is tissues arose here. Arose means evolutionarily speaking, this is when we start seeing tissues. And so we are now looking at the formation of bilateral symmetry. So that brings up a new group of categories that we need to discuss a little bit. The acoelomates, pseudocoelomates, and coelomates, and also the idea of protozomes versus deuterozomes. Now, I haven't shown you any pictures, but acoelomates are animals that don't have body cavities. So essentially their tissues are all just next to each other. Pseudocoelomates, you see different tissues, but they're not well, they might not be separated very well with connective tissue. And then in coelomates, you have true cavities that are covered with linings that separate the two. We also have what we call protozomes, which are going to be based on their embryology. The idea of protozomes and pro is when we start talking, we'll show some pictures, or I'll show you some pictures. When we talk about embryo development, protozomes, when we have a cell, gets fertilized, remember what that was called, a zygote. The eggs are dividing, or not the eggs, but the cells are dividing after being fertilized, and eventually you got this big sac of cells that are called a blastocele. Well, a hole forms, and that hole becomes the mouth of the insects or animals. It doesn't necessarily have to be an insect. Those are what we call protozomes. In the case of deuterozomes, that hole actually becomes the anus first. And so that is important because it tells, again, it's another tool for grouping animals that also says something about their evolutionary history. So the vast majority of animals are actually protozomes, where when they're an embryo, their mouth forms first. So that suggests that they have an evolutionary history that's separated from the deuterozomes, which include ourselves. We're a deuterozome. So I'll get into that a little bit. And then we talk about these animals down here being without segmentation. And then over here, we see animals with segmentation. Even we have segments. The easiest place to see it is if you have abs, because you didn't have fat covering them, you can see the segmented muscles. And obviously, insects, you can see segments, or earthworms, you'll see segments. You can see constrictions. and repeats of body segments. So that's what this is here showing you, is another example of the evolution of animals. It's not unlike what you're seeing here, but it's another diagram, or phylogenetic tree is what we call these, phylogenies, showing their evolutionary relationship. And so over here we have our sponges, which are porifera, and our common ancestors would be those canocyte protists. Remember, the protists are like from the group of amoebas. And then as we branch off this evolutionary tree, we have something that says a blind gut two-cell layer. This must be in reference, obviously, to their body anatomy. But why do we have a blind gut? Remember, in the case of jellyfish, they have the same gut in mouth. So the food comes in and goes out the same hole. And then we start to see animals with a complete gut. And when do we first see that? In the comb jelly. So remember, Centophora is the comb jelly. So it has a tube that goes all the way through, which maybe suggests a little bit of an advance in evolution. Again, advance is a slippery slope. I mean, sponges are great at being sponges, but we see this new evolutionary change in the anatomy of animals, and it seems to become with the formation of a complete gut. And then we start to see different, remember, with the jellyfish, we had two cell layers, an epidermis, which was the ectoderm, and an endoderm, which is the gastroderm. And then we had some kind of jelly matrix in the middle. Well, we have three true cell layers now forming. So we'll get into this in another picture, but we have basically an outer layer, which is the epidermis of ourselves or other animals like insects. Then we have a mesoderm, which is like our muscles. And then we have an endoderm, which is our digestive system. So we start to see three cell layers in embryologically and ultimately forming these layers in animals. So these are evolutionary advancements where you see these red dots. And so the protozoans are up here. We have Loaf. Remember, these are the animals that the mouth comes first in the embryo. And ectodisones are basically animals with a cuticle like insects, and so they have an exoskeleton. And then we have these trocazones, which include things like flatworms. So we'll get into that in a little bit. So those are examples of evolutionary changes. And then you start to see, so here it says a complex skeleton active locomotion. That's where you're starting to see that in these animals. And then you see internal skeletons for animals that are deuterosomes. Remember, insects have an exoskeleton, and the other animals we'll talk about, but we actually have a skeleton that's inside our muscles. That's an endoskeleton or internal skeleton. And that's part of the animals that represent deuterosomes. Again, a lot of deuterosomes include things like ourselves and birds and cats and reptiles and amphibians and fish and starfish. So we'll get to that. But anyway, that's another evolutionary change. And so I've just kind of described this already through showing you that picture, but basically we're going to talk about the different tissue layers of an animal that forms and what they become. Like, for instance, the ectoderm becomes, in us would be, or in other animals, a lot of animals, it would be where the nerves are found, and the endoderm is where the digestive system was found, like in the case of jellyfish. But we're actually triploblastic, and more advanced animals are tripoblastic, as I just mentioned in the previous slide also, where the mesoderm becomes muscles and other tissues. And so that's a big advancement. So the existence of three cell layers distinguishes the protozomes and the deuterozomes from simple animals. So jellyfish and sponge don't have those different layers. I don't believe, we might need to check on flatworms, but I don't believe they do either. So that's an example of a protozoan that still has these 2 layers. So anyway, we're going to go explore these different things, and it'll help you to understand how we group the animals on this chart here. So here's the protozoans, deuterozomes. So all these animals that are here are protozomes. So again, the protozomes are animals that when they were an embryo, when the egg was fertilized and the cells were dividing, the mouth developed into a blastopore. Anus developed later, and that was a deuterosome. And so that's ourselves, like, as I keep repeating myself over and over. But anyway, let's look at how the protozomes embryo develops. And again, these are animals that are mostly invertebrates. You have a single cell, a sperm fertilizes the egg. That single cell becomes fertilized. It is then known as a zygote. And then 10 or 12 hours later, or a few hours later, it just depends on the species, but that fertilized egg then starts to divide into 2, and then it divides again into 4. And it does so through a process called mitosis. And so then that 4 cell divides into 8 cells. But notice something kind of strange here. The division isn't equal anymore. So these divisions are relatively equal. We had equal sizes of the cells. In this case, we get unequal. We get 8 cells, but they're no longer equal in size. See how we got the top layers are very small cells, and the bottom is really big. And then they divide again, and we have 16 cells. And then we have 32 cells. And we actually start to call this spiral cleavage, because you're starting to see kind of a spirally shape form as the cells continue to divide. Now, in the case of deuterozomes, we see something kind of similar initially. Here we have our egg. A sperm has fertilized it. The egg divides into 2 equal halves. It divides into 4 equal halves. It then divides into 8 equal sizes. And you notice they're just equally on top of each other for the most part. So we call this radial cleavage. And then here we divide again and so forth until we have up to 32 cells, and they're relatively on top of each other. See how the colors are the same? That means those cells came from that group. So going on to the next page, this is again the protozomes up here and the deuterozomes down here, and we have spiral cleavage. But eventually, as the cells continue to divide, we have essentially what seems like a basketball. It's just a hollow, it's not, well, maybe it might be some fluids in here, but it's a hollow hole with cells. We call that a bastula. And then this kind of goes in dents, and we have what we call a blastopore. Now remember, the blastopore is the hole that will ultimately become the mouth in protozomes. And we also begin to form other new cells that form up the mesoderm. So this is a triple blastic animal, meaning it has 3 different tissue layers. The outer layer is the ectoderm, the inner layer is the mesoderm, the middle layer is the mesoderm, and then the inner layer is the endoderm. And then this continues getting bigger and bigger until the anus forms. So this is the tube that'll go throughout the animal's body. In this case, it's probably, well, let's say it's an insect. It could be a variety of different animals, but this would be how the embryo of an insect may have formed. Now we're down here, we're the deuterozomes. Let's go back. And here we are, the deuterozomes, and that includes animals like my cat that's sitting on my lap. The inside here of the blastula is this kind of like what we see with the insect. And then a hole forms, and that hole becomes the anus. So the blastopore of a deuterosome becomes an anus. And again, remember, this is radial division. I mean, when the cell divides, that's the division. The red, the mesoderm, so this is also a triple blastic animal, forms the muscles and so forth. So typically in animals, the ectoderm, and it doesn't matter which animals, but typically the ectoderm represents the outer layer, which would be skin and us, hair. Nervous system is actually made up of the ectoderm, if I recall right. And insects would be it wouldn't be their skin, it would be actually like their cuticle, because remember their skeleton's on the outside. And the mesoderm would become muscles, heart and stuff like that, visceral. And then the endoderm usually becomes the digestive system. So this would be representing like the stomach and the intestines and so forth. So that's where a lot of the fate of these cells will ultimately go. Regardless if it's an insect or a human, a lot of that basic rule applies. So again, deuterozomes and protozomes have some fundamental embryological differences that led to different evolutionary directions. Protozomes are eggs that cleave and do so spirally. Deuterozomes do it radially. And as you know, the blastocele and the blastopore become the anus in the deuterosome and the mouth in the protozome. This is kind of just tells you what I just had mentioned already. But the protozomes and the deuterozomes that dominate today's fauna, so most of the animals that dominate the fauna, meaning what animals of this time period, have been evolving separately since the Cambrian period. So it's been a long time since protozomes and deuterozomes have evolved from each other as an animal group. And then they went on and specialized and became vertebrates that crawl out of land or fish or whatever. Both lineages are bilaterally symmetrical. So that means you can split an insect in half and I'll have a left half and a right half. You can split ourselves and we have a left half and a right half. Again, there's many traits that are similar, like the central nervous system consists of the brain and the entrance to the digestive tract. That's true for both of us. Insects have a ventral, and when I say insects, I mean it kind of universally for a lot of those animals that are protozomes. But the nervous system is actually on the front side. While the spinal cord, the main nerve ganglion are going down the front side of a grasshopper or a caterpillar if you dissected it. And again, I'm meaning that kind of universally, there's some truth to that. There's always exceptions and stuff like that. For us, deuterozomes, the nervous system, the main part of it, central nervous system, travels down our back. So the brain and our spinal cord and things like that. So those are kind of the big differences. The major Okay, so let's look at this again. So talking about the evolution of bilaterally symmetrical animals, protozomes and deuterozomes was a big split. And then, as I mentioned already, deuterozomes have a dorsal nervous system. It says larvae if present, because we don't have larvae running around. They can collect food using a single cilium, which allows them to I guess, push food into their mouth. But again, we'll get to that when we get to that. A blastopore, as I mentioned, becomes the anus. Radial cleavage, I've already mentioned that. Evidence suggests that the protozoa split into two major lineages that have been evolving separately since ancient times. So remember, the protozoans include things like our flatworms and our insects and our arthropods and all sorts of things. Those are the ecdysones. So when we talk about arthropods, they increase in size by molting their external skeleton, or if we're talking about crabs or lobsters and things like that. And they have a group of genes called homeobox genes that are involved in that. And so the other animals that aren't with an exoskeleton in the sense of like an insect or arthropod, those are known as Loaf trocazones, and they grow by adding the size to their skeleton elements and using cilia for locomotion. That means they have little hairy-like movements that help them to move around. So these would be things like flatworms. And then they will have larvae running around called trochophores. So anyway, we'll get into these in a moment, and then we'll get into these animals, and then it'll make a lot of sense when you look back at this chart here. So like these would be the animals that have the ecdysones, for instance. Now, looking at the body cavities, remember that was a big thing under our heading also. I'm sorry, jump back one more. So we're talking about coelomates now, pseudocoelomates, and coelomates. So these are all important evolutionarily. And so you can see as you follow up this chart, body cavity, we have pseudocoels and coeloms. And so the pseudocoeloms include these things like these worms here. And there's lots of different worms because we have the earthworms over here, and over here are the nematodes. And then we have these rotifers. And so those are animals that have what they call a pseudocoel. While the flatworms over here, Platyhelminths, are acoelomates, so they don't have those coeloms, they don't have those body cavities. So you can see body cavities evolved here. And then if you look at coeloms or true coeloms, that means they have true body cavities. That includes all these animals going up above it. So that's how we categorized them. We start to see mollusks, annelids, arthropods, and then some animals we don't recognize very easily. And then we see starfish and then chordata, which include ourselves. Again, what's happened is people that have studied animals looked and studied their anatomy, started seeing these trends, or embryology, and started seeing these trends. And then that's what developed these kind of phylogenies, these evolutionary trees. And it gives you an idea of how closely related each of these they are to each other. So going back to our body cavities again, the flatworms don't have true cavities. They still have the three different tissues. So we have an ectoderm, an endoderm, and a mesoderm, or we call it mesenchyme. But there's nothing really separating them. So fluids can easily pass through them. There's no connective tissue separating them. So there's not true cavities. Now, pseudocoelomates have what they call a pseudocoel, which is a liquid-filled space which the organs are suspended, but they're not completely enclosed from each other like a true cavity. If you go over here and look at this earthworm, so this is a roundworm, which is a type of nematode, and compared to an earthworm, which evolutionarily you could argue is more advanced. And again, in quotes, because what is advanced depends a lot on what you're trying to argue. But this coelom trait comes later, and you can see there's an actual covering that makes for a true body cavity. And we have our three different tissues, and now they're more clearly separated from one another, and the fluids are more easily floating between them. Some of the simplest animals actually move through the use of their hydrostatic skeletons. That means that they can move fluids and help them push out tissues and help them to stretch out. So they can squeeze, push fluid forward, and actually push their body forward. So we're using muscles, obviously, to move my bones and muscles. They're actually kind of like if you had a water-filled balloon and you squeezed one end, the front end would get expanded and move forward, and then that would actually help move the balloon forward. Well, animals can actually do that for locomotion. They can also puff up their body to make their size bigger and then form a new exoskeleton. That's how insects molt, for instance. So there's all sorts of hydrostatic kind of stuff that goes on in these other animals that you don't see in deuterosomes for the most part. Again, there's always exceptions probably, but that's how a lot of these animals move, or they have little cilia that help them to move. So here's another look at groups of animals that are coming under the Lofa trocazones. So that means animals that so this is just another phylogeny that's focusing on Lofa trocazones, which include animals like flatworms and rotifers. The next group of animals we'll start to talk about, bryozoas, brachiopods, phorninidae, nematodes, annelids, and mollusks. Then we'll start talking, that's not on this chart, but if you go back to that the first original phylogeny I showed you, then we'll start talking about ecodisones, which include insects and arthropods, and then ultimately deuterosomes that include ourselves. And so this is just showing you some of the evolutionary adaptations that we see in the formation of these ancestors, where we have embryonic cell layers, 3 of them, so the triploblastic. You'll see a thing called a lophophore that for these animals here. And a lophophore is going to be like a tube that goes through it that you'll see a little bit down the road when we talk about it. It's also going to talk about, again, these are just evolutionary advancements. Here we have a cleavage, and then we see a segmented body plan form for annelids. So now we're going to get into animals that are bilateral. So remember, we went from sponges, no symmetry, to jellyfish with radiosymmetry, and then basically the rest of the animals will have some type of bilateral symmetry. And we've already covered this extensively, so I'm not going to spend any time on this other than to show you it, this picture. Here we see a bilateral animal split down the sagittal plane, and then that gives them a left and a right half. While if we cut down the frontal plane, then it'll be a top and a bottom. So the top half would be called a dorsal side. The bottom half would be called a ventral side. The front half is called the anterior, and the tail region is called the posterior region. So that's how a bilateral animal would be divided, and that's what we're going to start talking about next. Before that, animals were radiosymmetry, where you could slice them in any different direction, and they wouldn't necessarily have a true, they're not going to have a posterior half or not. It's just going to be, it'll look the same almost any direction you cut it. And of course, with bilateral symmetry, we see cephalization. If you remember that, that is the head region with eyes and mouth and so forth. So the first group of animals will be our flatworms, which were acoelomate. Remember, they don't have any true body cavities. They do have the three different tissue layers, the ectoderm, the mesoderm, and the endoderm. And they have kind of very primitive eyes that can help with picking up on light. And so now let's take a look at flatworms, and there's a variety of them. We do have a nervous system that's found up from the skin and ectoderm and so forth. So the platyhelminths are the simplest of the Loaf trocazones. They're known as flatworms. Flatworms are bilaterally symmetrical, but very, very flat. They're unsegmented acoelomates. They lack organs for transporting oxygen, so that means oxygen is going right through their skin and into their internal tissues. They have very simple organs for excreting metabolic waste. So they have what we call, they wouldn't be much of, they wouldn't have really a true kidney or anything like that. And a lot of the waste would just actually be released via their skin. So they're very flattened in shape. And so when they're very flat like that, it helps them to diffuse oxygen right into the tissues from the environment. The flatworm has a digestive tract that is a mouth that's opening into a blind sac. The sac is often highly branched, increasing the surface area available for absorption of nutrients. So that everything you'll see in biology, the surface area increases the opportunity for collecting oxygen or for digestion. That's a big thing in biology and physiology. Flatworms feed on both dead or living tissues. When they feed on living tissues, they're often parasitic. So you can actually find them as a parasite in your body. But there's also free-living flatworms in the ocean. They move by beating on a band of cilia. Remember, cilia are going to be like little hairs that are found along their side of their body or underneath their body. So here is our flatworms. We have our platyhelminths, which is our largest phylum of solid worms. So here's our flatworms. A lot of times, I believe you can cut them in half and they'll actually reform. They don't necessarily die. They're also hermaphroditic. That means they have both male and female sexual parts. And so they can actually, hook up with each other, impregnate each other, and then they go off and have offspring. Again, a lot of them are free living, but there are some that are what known as tapeworms and flukes. And these are the parasites that can live inside bodies. Again, they lack a true circulatory system, so they're not going to have blood vessels and arteries and things like that. So here's an example of how it can be a parasite, the non-free living ones. Here we have this guy who has encountered eating raw fish or other animals, might have done it, other mammals. And so they've eaten the raw fish, the eggs go into their body as adult flukes, and then it'll sit on their liver or some other portion of their body and live, and will put eggs into their digestive system, and then it'll be pooped out. A snail will eat the eggs, and then the eggs will actually break open inside the snail, and the larvae is known as well, not necessarily a larvae, but the first stage is known as a mericidium that hatches after being eaten by a snail. This becomes a sporocyst. So this mericidium becomes a sporocyst and continues developing, and then it's known as a radia. And then it can also be known as a cercaria, which can actually swim and not impregnate, but penetrate the muscles of fish. And then it forms a little cyst inside the muscles of fish, known as a metacercal cyst. And then that's what the human has eaten in this particular picture. He's eaten raw fish, breaking open the muscle, and then the metacercal cyst encounters the digestive system or whatever. But once it gets into the body, opens up as an adult fluke and will live on its liver or some other organ. And so here we see the adult fluke. So this is the flatworm here. So you can see its mouth attachment here. It, remember, has male and female sexual parts, but it'll need to hook up with another one usually to impregnate those eggs or to fertilize those eggs. So here's maybe some ovaries and testes. Most living flatworms are parasitic, such as tapeworms. Cestozodia is the name, is the class, Cestozodia. Remember, kingdom, phylum, class, order. And flukes are known as Traumatatodia. If you take a parasite class with Dr. Meager, you might get to learn more about them. But the parasitic flatworms lack a digestive tract and they absorb their digestion from their host directly, so they can absorb the nutrients. Some species cause some serious diseases. Most parasitic species have complex life cycles involving one or more intermediate hosts in several larval stages. So again, it's an incredible life cycle. Look at that. This person had to eat a raw fish or eat some fish that was a little raw, broke open these adult fluke that hooked onto them. Eggs were dropped off. A snail ate the eggs. The larval fluke developed and then swam out of or was eaten, depending on the case, by the snail, swims over and hooks up with a fish. So that's a really complex life cycle to have these 3 different organisms that it had to interact with to become adult again. And anywhere along the line, if that fish had died and not been eaten, those animals would have never made it into the human and would have died also. So it is incredible to see these complex life cycles that have evolved with these different parasites. Looking at the anatomy of this flatworm, you can see it has both testes and eggs, as I mentioned before, ovaries. And here's the vagina and egg, and it can basically connect onto a liver, for instance. Here's another example where we have this tapeworm in this case, and it has eggs that were pooped, or well, maybe pooped out. Let's see. We have a larval stage, and then it hooks up with this, what we call a copepod. And the copepod is like a invertebrate that's found in water that has a little bit of a hard shell. So it's kind of, I won't call it a shrimp, but you got the gist. It's just a little shrimp-like animal that other fish will eat. So the larvae, have been pooped out, hook up with the intermediate host. The fish eat it, and then the bear eats the fish, and then this larvae now becomes an adult tapeworm or mature tapeworm that actually will hook up onto the intestines of this bear or human or other animals and form new segments. And each one of these segments will then can be pooped out and continue this life cycle. So again, it's a very complex life cycle. Looks like it might have even gone through 2 different fish species before it was eaten by a bear. So again, really crazy. Here is what a tapeworm looks like. You can see the head region, and I'm putting head in quotes, has hooks that will impregnate, or not impregnate, I'm using that word too much, will penetrate into the intestines of a mammal of some sort or other animal. Dogs have tapeworms and cats, you give them, and when we talk about giving them deworming medicine, if you pick up a pet from the pet store or from the animal clinic, they give them deworming medicine. It's to get rid of these tapeworms often. It could be nematodes too, but often tapeworms. And so here it is, hooks up. And again, a tapeworm is a type of flatworm. The hooks will get into the intestinal wall, and then repeating segments of reproductive parts will form. And again, it's absorbing nutrients from the intestine. So what you're eating is now their meal too. That's how it works. And these proglottid segments then can break off, form a new head or whatnot. And then ultimately I'll go through the life cycle, what's pooped out, and we go back to that previous slide and we'll have this life cycle for tapeworms. Now there's also flatworms that are not parasitic that are found on the ocean floor. So here's an example of a flatworm in the class, not family, kingdom, phylum, class, Turbillaria. And these are the ancestral flatworm forms. They're small, free-living, marine, freshwater animals. They have a head with chemoreceptors, organs, simple eyes, and a very small brain. And then if you watch some animal documentaries, you'll see these occasionally floating around on the seafloor. All right, so I think this is a pretty good place to pause. You'll see I'll probably put a little homework video that talks about tapeworms and flatworms. Again, some are free living like this one here, living in soil, but others are parasitic. Most of them are parasitic. Some are free living, living in oceans. They have a very flat body. That's the main thing. They have a very flat body. They're acoelomates. They absorb oxygen primarily through the air, or waste through the air, absorb nutrients through their body. They're just so thin and flat, that's how they can get away with that kind of stuff. So that's the main things to get from this section. Go ahead and write a half-page summary on what you learned and turn it in, and I'll get on to the next lecture. Take care.